Good evening, and welcome. My name is Kevin Fitzgerald, and I am chair of the Department of Medical Humanities at Creighton University School of Medicine here in Phoenix and in Omaha at both our campuses. And we're very excited to welcome you this evening to the Dr. Lori Schweickert Endowed Lecture Series event. Dr. Schweickert, who is here with us this evening, has generously endowed this lecture series so that we can bring up topics that are at the forefront of where medicine may be going. And these advances raise both scientific and ethical challenges for us as we move forward. And so this evening, we are very pleased that Dr. Schweickert, in her Creighton history, has decided that this was something that she wanted to make happen for our students and for the public. Now, Dr. Schweickert graduated undergraduate at Creighton University and also graduated from the School of Medicine there. But you may not know this, but her most famous contribution to the Creighton University was that she was the first, were you the only? Betty Blue Jay. So we have a mascot, the Blue Jay, for our basketball and sports teams and all, and it's usually Billy Blue Jay, but for one time, it was Betty Blue Jay, and that was Dr. Schweickert. So she stands alone in history as that contribution. But in addition, she's had a marvelous career as a child and adolescent psychiatrist. And as part of her desire to give back, to carry out that Creighton legacy that she picked up in the School of Medicine and as an undergrad, she also went on multiple medical missions around the world, and then also wanted to do something more for our students, to have this opportunity to address these cutting-edge questions that are before all of us. And so just to begin, I'd like to give an opportunity for Dr. Schweickert. If you'd like to say a few words, we have a microphone for you. Come over here a little bit. There we go.
Good. Well, first of all, I wanted to say what a privilege it is to partner with Creighton University on this endowed lecture series, and to thank the people who really shepherded this from the beginning, which is Father Kevin Fitzgerald and Chris Otepka. So this was born really of watching good clinical, good clinicians, people of faith, really struggle with the ways of bringing God and faith and into the difficult conversations and decisions that they have to make as clinicians. And so if this brings that to the forefront and lets people think and ask questions and have dialogue around this and educate themselves about this, that's what this lectureship is for. And so I'm very grateful for those here in attendance and those watching remotely, and we'll get on with it.
Thank you so much. Thank you. So we'll move on to introducing our two presenters this evening. To my right is Dr. Sadik Kassim. He is the Chief Scientific and Chief Technical Officer for Omic Solutions at Danaher. Danaher is a global life sciences and diagnostics innovator company. And he has himself a PhD in viral immunology. He specifically studied and published on innate immune response to herpes simplex virus type 1 and influenza virus. So he's an experienced scientist and executive with a track record in the biotechnology industry. He has worked on AAV-based gene therapy, immunotherapy, oncology, CAR T-cell therapies, T-cell therapies, CRISPR gene editing, hematopoietic stem cells, and CMC. And in addition, at times he finds the opportunity to sleep. Today he is with us, and I just found out to celebrate the 1-year anniversary of baby KJ Muldoon's genetic treatment. And Dr. Kassim was involved very intimately in the process and was leading the Danaher contribution to that amazing advance in medical treatment. To his right is my good friend Tim Hunt, who is CEO of the Alliance for Regenerative Medicine. He has a BA in History and Philosophy from Boston College and received a JD from the Columbus School of Law at the Catholic University of America. Now, the Alliance for Regenerative Medicine, if you're not familiar with it, is an international advocacy organization championing the benefits of engineered cell therapies and genetic medicines. Tim joined them in 2022 as CEO, and he has more than 20 years of experience in the biotechnology industry. So together tonight, two of the people who are at the forefront of this incredible advance in genetic medicine. What we will do to start is have Dr. Kassim give us a little ground information and some insight, both scientific and ethical, into the baby KJ Muldoon experience. So here's your clicker and go for it. Thank you.
Thank you, Dr. Schweickert, for creating this forum to discuss such topics. And thank you, Tim, for connecting me with the team here at Creighton and with Father Fitzgerald. So what I want to do over the next 10, 15 minutes is walk you through the story of baby KJ in 3 sections. First, give you a high-level overview of genomic medicine. What do we mean when we talk about genetic medicine? Then go into the details of baby KJ. As Father Fitzgerald mentioned, this is the 1-year anniversary where he received that drug infusion on 2/25/2025. And then conclusion and future direction, some of the questions that arise from the treatment of KJ. So what is genomic medicine? At a high level, genomic medicine is essentially all of the genetic information that's encoded within your cell. So every cell in your body has a code, that code being the code of life, 3.2 billion base pairs, A, T, C, G, and they encode for many of the features that make you unique. Your hair color, your height, your personality to a certain degree. If we were to take those 3.2 billion letters and put them in Times New Roman 12 font and then put them on a page 8.5 by 11, the stack would be higher than that of the Statue of Liberty. And that's what this, so the book of life, a lot of information encoded within each of your cells of your body. Now, what do we know about the genome? Well, we know that this book of life encodes about 20,000 genes, of which 5,000 are directly linked to disease, but 10,000, we still don't understand what their function is. So we've mapped out the book of life, we've written the book of life, but we don't necessarily know what each gene affects and what diseases emerge from these particularly affected genes. Now, the major problem in the field of genomic medicine relates to these 3 lines that I've plotted here. So on the blue line relates to the price of genetic sequencing. So genetic sequencing is that activity of trying to read the book of life. When the Human Genome Project started, it took about 20 years and $1 billion to sequence a single individual's entire genome. 20 years, $1 billion. In the last 20 years, we've seen the price drop from $10 million to now, last week it was announced $100. So you can now sequence the entire human genome in about $100. Now, compound that with the speed of how quickly we're able to do it. So it took 20 years for the first human genome. Current genome, we can sequence it in 20 minutes. The Broad Clinical Labs has the Guinness Book World Record for sequencing. This was announced last year. They took a sample, sequenced it in 20 minutes, and then from end to end, sample prep and annotating that genome about 4 hours. So this is the age that we're living in. Now, this has led to an increase in the number of genetic diseases added annually. So remember, we have 10,000 genes that we don't know what they function. as we sequence more genes, we now understand the function to genetic outcomes, to pathological outcomes. And we're adding roughly about 250 genetic diseases to our compendium per year. Now, this is where the problem is and the opportunity. The number of therapies that have been developed for these genetic diseases lags woefully behind. So there's a huge gap there. And when we look at genetic disorders, many folks think that genetic disorders are rare disorders. And by rare, using the colloquial meaning that infrequent. But the reality is that genetic disorders, individually, they may be rare. So a certain genetic disorder may affect 10 or 15 individuals. But collectively, if you take all of the rare genetic disorders and aggregate them across the world, 400 million people are affected with rare genetic disorders, of which 30 million live in the United States. And if we extend it into all disorders like cardiovascular disease, neurodegenerative disorders, then it becomes every disorder that one is affected with has a genetic component. So by cracking the code or figuring out the path from sequencing to intervention for rare disorders, we're really opening up a path for treating all genetic disorders or all disorders that have a genetic underpinning. Now I want to tell you a bit about CRISPR gene editing. CRISPR gene editing is one of the tools that we have in our repertoire that may enable us to intervene at the genetic level and really fix a lot of these mutations that cause pathologies. Dr. Jennifer Doudna and Emmanuel Charpentier received the Nobel Prize back in 2020 for inventing something called CRISPR gene editing. CRISPR gene editing at a high level is kind of like the copy-paste mechanism that you have on your word processor when you're writing a document on Microsoft Word. So it's an exceptionally targeted tool that enables you to specifically intervene at the DNA level and to make edits. And these edits could be removing a single letter that causes the genetic disorder, adding some new code, or swapping out code. And it's an exceptional pair of molecular tools that we have at our disposal today that enable us to really make an impact. Now, Danaher is a global life science and diagnostic company, as Father Fitzgerald mentioned. And so what we want, one of our motivations and objectives is to get these tools out into the world so that folks can make a meaningful impact in terms of healthcare. And these tools need to be in the hands of scientists, drug development companies, and other organizations that have human health as an objective. So 2 years ago, we partnered with Dr. Jennifer Doudna, who won the Nobel Prize, and Fyodor Urnov at the Innovative Genomics Institute in Berkeley to come up with a cookbook that enables us to use CRISPR gene editing to platform it across all rare genetic disorders. We partnered with the Alliance for Regenerative Medicines as well to really work on the regulatory front, because what CRISPR will enable is a complete paradigm shift in how we can approach drug development. And this next slide really describes it. The current drug development timeline for making a drug, it costs a bit of over $1 billion in about 10 years, roughly, for a new drug, like a Wegovy or an Ozempic or a Keytruda. So it's a long and path filled with a lot of failures and inefficiencies. But with CRISPR gene editing, because the basic payload can be swapped out between each genetic disorder, you don't need to reinvent the wheel. All you really need to do is change that genetic sequence that's specific for that patient. And now you have a modular format, a modular way to develop drugs where you don't need to reinvent the wheel each time, but just simply swap out the genetic sequence of interest and then use that to treat the patient. And so when we talk about a platform, and this is an important concept, what we're talking about here is that there are off-the-shelf components that can be leveraged across the millions of patients, but then there are custom components that can be customized for that individual. And then in that way, you can make an impact where historically drug development has not gone, because it's not worth it if you only have 10 or 20 individuals that are affected. And so now you have this flywheel. Rather than a linear path, it becomes a flywheel that enables you to treat the millions of patients with the thousands of genetic disorders that I mentioned. So this is where the story of KJ comes in. two physicians at the Children's Hospital of Philadelphia and the University of Pennsylvania have been working towards developing a gene editing platform for urea cycle disorders. What are urea cycle disorders? So in your liver carries out a lot of the metabolic functions of your physiology. So the liver helps with protein metabolism. Now, in a normal liver, there's the urea cycle that helps to break down the individual protein into individual amino acid components and therefore give you the nutrients that you need. In a liver that's affected by a urea cycle disorder, there's a mutation in one of these 8 genes, and that mutation causes an inability to metabolize protein. And what ends up happening is you get an elevation of urea, which is a toxic metabolite, ammonia, in the circulation. And that leads to developmental disorders and potential death. The only real treatment for the most severe types of urea cycle disorders is a liver transplant. And currently, there aren't really a lot of treatments here, because if you look at the number of mutations that across these 8 genes, there's approximately 500 mutations. And so to really develop but each of these mutations maybe collectively only affects 1,000 patients or maybe 2,000 patients. So economically, there's no incentive to develop a drug. And this is where if you can platform it across the 500 mutations, you can potentially make an impact for the 1 million patients that have these disorders. So baby KJ was born in August of 2024. And immediately demonstrated a lot of the symptoms of someone with a urea cycle disorder. He had to be put on immediate life support and on the standard of care, which is scavenging therapy to remove the ammonia from his circulation because it gets to really high toxic levels. He was lucky and fortunate to be born in a hospital that had access to next generation newborn sequencing. So immediately the OBGYN recognized these symptoms and said, we need to sequence his sample to see if this is truly a urea cycle disorder. And rapidly what they were able to identify is that KJ inherited both a mutation from his mother's side and his father's side that caused this urea cycle disorder. And he had one of the most severe types of mutations, which is in a gene called CPS1. And this is just the cycle of what's affected. And he literally had for someone with this mutation, the prognosis is death within the first week of life. They were able to maintain and stabilize him with some of the standard of care therapies, which prolonged his life. But this is where Becca and Kieran reached out to us at Danaher in partnership with the IGI and said, look, we've identified this mutation, we've identified the sequence, now the clock is ticking. We need to manufacture this drug. We don't have the capability to manufacture it. Danaher, can you help collaborate? And we led the effort to manufacture the drug product. And this really took an entire village. I'm not going to go into the full timeline because it's published. But from the time of the genetic diagnosis to running some of the basic preclinical studies to manufacturing the drug to the infusion, which occurred a year ago today, February 25, it was a total of less than 6 months. And we, discussed with the FDA as well. So how's KJ doing today? So this is KJ last November at an Eagles game wearing his jersey. And this is KJ on the stage in Boston last December being carried. And now there's footage of KJ walking, taking his first steps. He's over a year old. And by all measures, he shouldn't be around. But his story demonstrates the power of being able to rapidly sequence, diagnose, intervene in a meaningful period of time to really save a life. So what are the implications of baby KJ and how can we treat more patients? Well, the case report was published in the New England Journal of Medicine. It was really expedited in terms of publication, and the reviewer comments are here. I mean, I think the consensus is that the framework that was enabled by KJ really, establishes a roadmap for how to do this for many other patients. And this was picked up by the Secretary of Health and Human Services, who then, announced funding for research to enable more types of therapies like KJ. And I think this brings into mind, I know at Creighton, very much underpinned by the Jesuit values and by the Catholic tradition. And I think from the Judeo-Islamic tradition and the Abrahamic tradition, I think the case of KJ really highlights the Talmudic saying and the Quranic saying, whoever saves a single life, it's as if he has saved an entire world. And I think the aspiration here is, can we recapitulate this roadmap, not just for KJ, with a lot of luck and good fortune, but can we systemize and institutionalize this so that any individual would have access to this type of a therapy? But there are some outstanding ethical questions that we may get into. And actually, a lot of these outstanding ethical questions were highlighted by The Economist when CRISPR first came to the scene many years ago. The Economist of all places, highlighting that the only way forward is ethics in terms of intervening and editing humanity. And I think these are some of the questions that I flagged. And obviously there are many more questions, but like, how do we ensure equitable access to rapidly developed genetic medicines, whether it's gene editing, AAV gene therapy, cell therapy? Tim will talk about some of the advocacy work that ARM is doing to ensure access, but this is an important question, especially when some of these diseases affect areas with less advanced infrastructure. Standards of informed consent. What do we need to take when urgent decisions need to be made? So if you have a life and death situation and a newborn, the parents are going to be, need to be, messaged the appropriate message. And then I think the final question that I'll highlight here is, how do different religious and secular traditions interpret humanity's role in altering or repairing the human genome? And is it considered healing or overstepping human moral authority? And I think that's the bigger metaphysical question. So the team of KJ was an entire army. I think another thing that I want to highlight is just the extreme collaborative nature of this project. And I think the roadmap is still being defined, and we need more of these collaborations between different institutions and different players that don't typically collaborate with each other. And then these are the folks that are working with us at Berkeley and Danaher companies. Thank you for listening.
Wow, thank you.
Thanks.
Thank you, Dr. Kassim. That's spectacular. Wow. That must have been incredibly exciting to be part of that endeavor. That was amazing. Absolutely. But of course, as you say, it leads us to those bigger questions. Where do we go from here? And for that answer, we're going to turn to Tim Hunt, who is going to lay out the future for us a little bit in terms of this industry and the companies that are involved in it, and what they see and where they hope things go.
Correct. Yeah, so my talk is on some work we've done within our organization on cell and gene therapy ethics and society, and it's a lot of building on what Sadik was talking about. Like, so if you roll back the clock almost 10 years ago when I was at this company, Editas Medicine, CRISPR gene editing, I remember that when that Economist article came out. I got to know Kevin around that time. When you look at this technology, it's highly disruptive, right? It's very different than in the bigger world of pharmaceuticals and biotechnology, a small molecule or a standard monoclonal antibody or other things, an oligonucleotide, right? It kind of gets at these tricky questions, and I'll get into that in a second. But some of it really kind of arcs up into when you get down into some of the applications, like what does it mean to be human? So you have to think about a lot of these things differently, and I'll get into some examples of how we think about that. So the Alliance for Regenerative Medicine, as I said, we have we're a nonprofit organization. We're headquartered in Washington, D.C. We do a lot of work in Europe and the U.S., a little bit of work in Asia, starting to do a little bit of work in the Middle East. And we're made up of organizations, anything from small private biotechnology companies to public biotech companies to some of the large pharmaceutical companies. And the key thing that binds us all together is we're only focused on cell therapy, gene editing, and a little bit of tissue engineering. And then we've got a few dozen of the major academic centers that are focused in this area. The Mayo Clinic's a prominent member, UPenn, organizations like that. Sadik was talking about the team at Penn. And then we've got a few dozen patient organizations, mostly rare disease patient organizations, and then a lot of the companies that make the stuff and enable the field. Danaher is one of our prominent members, and other organizations. So it's a very diverse community, and we're focused on a number of things, right? We convene the sector. We have 2 big meetings a year. Our big annual meeting is out in Arizona every year in October, so you're all invited to come in October, first week of October, the 5th through the 7th. And those that are watching us online, please come to Phoenix. We have our big European meeting taking place in Rome at the end of April. We put together a lot of data and analytics about the field, and we're very focused on engaging key stakeholders to think about how do we try to modernize these healthcare systems. Think of like regulators like the FDA and the European counterparts, and payers like Medicaid, Medicare, and European payers, so they can embrace these very disruptive technologies. Because a lot of these systems were built in the 1960s and 1970s around things like small molecules that my grandparents used to take, right? So it's a lot around our efforts to try to modernize healthcare systems. And we really try to think of it as the patient is our North Star. So people like children like baby KJ, right, are our North Stars. And I showed this slide at a investor conference in January, and we thought about it a little bit. We were like, there's really some amazing bookends that you can think about, and actually that took place last year. So baby KJ was dosed last year, as Sadik indicated, right? That's a great example of, really a heroic effort by many, many teams, as Sadik pointed out, to save the life of a patient, right? Sort of an extreme example of where time is life, right? On the other end, a very, very hot and upcoming field are cell therapies they've been in CAR T therapies have been in liquid tumors for many years now. But a very promising area is where we might be able to intervene for autoimmune conditions. So think about refractory patients that have lupus or multiple sclerosis or other tricky scleroderma, other tricky areas. And there was a woman named Marcy McHugh, and last October, she came to our meeting on the Mesa here in Arizona, and she spoke about her particular experience with refractory multiple sclerosis. And she said, I don't have to worry that I'm going to wake up tomorrow and lose function. That fear is not there anymore. And that's the other extreme, right? It's a real thing for a lot of patients to lose functionality, right? They know the natural history. They can see they may have been a wheelchair or worse, right, over time. So these therapies can, when they work well, and they don't always work well, but they work well, it can be transformative at both saving lives and preserving functionality. Okay. Very busy slide, but this slide, we track, as I mentioned, we crunch a lot of numbers and have a lot of data. One of the things we do is we track the sort of global pipeline for cell and gene therapies and look at which regions are doing what. So for the first time, there's been such explosive growth in the Asia Pacific region that they have eclipsed North America in the number of clinical trials that are taking place. So this is a chart that reflects the number of clinical trials. We don't have the numbers up on Europe up here, but if we did, they'd be about half, probably a little bit less than half of the size of what you see in the United States, North America, right? So 300-400, something like that, right? So really, the 2 huge regions are Asia, led by China, and North America, led by the United States. And what we saw for the first time was, we can get into this more in the Q&A, the growth of China in this area. China grew by 20% in 6 months in the number of clinical trials that they have going on, which is really stunning and sort of a wake-up call for many other regions led by the US and Europe to try to get their act together to make things easier so that we can start early phase 1 clinical trials in these different regions. So a lot of global competition surging right now. Okay, let's get to the good stuff. So I mentioned how disruptive the technology is, cell and gene therapy is, right? And when I first started at ARM, I spent a little time with the team, the internal team, talking to them about, look, we need to think about our engagement model a little bit differently and think about some of our major stakeholders and some of the basic practical questions that they ask all the time. And these are like the kind of things people are like, we don't really want to talk about some of this stuff. It's better not to talk about the science. And that's something that I just learned over the years is lunacy, right? You have to really think about who are the people that you care a lot about and what's top of mind for them and lean into it and have some honest conversations, even if they're hard for you or for them, right? And so I laid out a few slides. I think I sketched it out like in pencil and had somebody put it up in PowerPoint, and then we've refined it a little bit over time. But I'm like, who are some of the people that we really care about, right? They're patients. I said we give them special names. We call them patients and caregivers, policy leaders, payers, right? Regulators, religious leaders, bioethicists, the media, investors, right? These are organizations that are really important at helping shape our bigger ecosystem, right? And guess what? They ask some pretty good questions. Some of them are tough, some of them are a little bit awkward, but they're important, right? They're very legitimate. You know, cell and gene therapies, cell therapies in the CAR T space can cost $400,000 to $500,000 in the United States, and gene therapies can cost on the low end probably $2 million, and on the more expensive end $4 million per patient. That's expensive, right? We need to be able to have that honest discussion. There's a reason why, but that's very expensive, right? Given that, can society, can these healthcare systems really afford these therapies, or will they? There's a lot of concern that, you know, what will happen? Will they overstress the system? Legitimate question. Will only the wealthy countries have access to these therapies? This is something Kevin and I've spent a lot of time with, right? It's not gotten lost on people that 80% of the world's sickle cell population is not in the US and it's not in Europe. It's primarily in a small number of African countries and India, parts of Asia. You know, with the Sadik was talking about baby KJ, and some of these disorders are very small in number. They might have 10 or a few dozen or a few hundred, even on the bigger side, 1,000 or a few thousand. That's not a great business model, right? Even at $2 million to $3 million a year, right? It's good, but if you think about how much money you might invest and how many years you might have to recoup that investment, it's not always a great business model. So is that model really built for the kind of commercial model built for these ultra rare disorders? It's a good question. How well regulated is the science? And then things like, what about, this is what I'll spend a little bit more time on, and I've spent some time with Kevin and some of his buddies over at the Vatican lately, and we'll be spending more time with them on this topic. You know, how do you think about abuses to the science, right? Where sometimes you have rogue scientists, and how do you see the world of genetically modified or designer babies? This gets into some of the more, you know, eugenic aspects of the technology, right? Things like enhancement versus treating someone that's not well. Or I happened to be by dumb luck in Hong Kong back in November of 2018 when the first genetically modified twins were announced in Hong Kong at what was the second international summit between the National Academy of Science, the Royal Society in the UK, and the Hong Kong Society of Science. It was a very kind of searing moment for those of us that were there. You know, the first time CRISPR was used to bring, in part to help bring what ended up being twins into the world, and later was announced it was a third child. So that generally widely considered to be an abuse of the technology. Okay, so we kicked off a project at ARM back in January, kind of a structured project back in January of 2024 that we called our Ethics and Society Project to grapple with, notice, grapple with, not solve, some of these thorny issues, right? Like, how do we think about cell and gene therapy pricing and value, and how do we get the balance right between innovation, which is really needed because the status quo for too many of our patients represents death or serious disability, right? You need to constantly innovate to try to save lives and improve functionality. How do we ensure that access is real, both in, yes, the United States and across Europe, but also other parts of the world? I'll get into that in a moment. And then sustainability, so that healthcare systems can absorb these powerful but sometimes expensive modalities, right? Second, how do we ensure that the further industrialization of the field, and Sadik did a great job talking about how the cost of sequencing has gone down so dramatically, right? We want to see a day where the cost of goods comes down dramatically, manufacturing costs come down, and we might be able to have more in vivo therapies and deliver them to many parts of the world that are very tricky to deliver to today. A lot of this generation of cell and gene therapies is logistically complex at times, right? So we want to eventually see that industrialization that will, hopefully lead to greater global access and democratization of the technology outside of just the developed markets. I mentioned the challenges of the ultra rare disease model. How does that play out over time? And then how do we think about what we call scientific engagement and ensuring appropriate guardrails around frontier technology? That's code word for some of this germline editing. Okay, let's drill a little bit more into the germline editing stuff. So this is really like a thing that's out there on the kind of really frontiers of society, right? Francis Collins, the longtime head of the NIH, once called germline editing a red line that should never be crossed. And yet, as I mentioned earlier, we're aware of at least a couple of occasions where this red line has been crossed, and there's always talk of both sort of academics and now kind of company people, biotech startup-y people, and some Silicon Valley investors that kind of are flirting with, wow, maybe we could do this germline editing sooner rather than later for a number of purposes. And so if you go back, there was an article that was written up in Nature that talked about polygenetic embryo editing that could play a role in preventing some disorders. This was sort of a speculative, in the years, in the next few decades, maybe this is something that could happen. But that got a lot of people kind of rattled, right? Understandably. The Wall Street Journal was reporting, and a lot of other publications, about this Dr. He, this scientist from China that I mentioned, who ended up in prison, was sort of back. He's now trying to round up other funding and doing other kinds of other work. And then there's these startups that have sort of emerged out of Silicon Valley. There's a number of them that have really appeared that are trying to raise money and are kind of pushing the envelope on germline editing, trying to do early studies, and eventually hope to get laws overturned in the United States and other countries to allow this to take place. So with that backdrop, there was a great deal of consternation in our community, understandably, both from prominent academic leaders and industry people that, we probably want to, someday, someone's probably going to come up and say, well, I've done it again, right? Maybe in a lightly regulated country, they're going to say my technique was better. I can do, they've got sort of the scientific arrogance that comes with it. So we decided that we were going to partner with ISCT, the International Society of Cell and Gene Therapy, and ASGCT, the American Society of Gene and Cell Therapy, to do a convening in March of last year. And we brought together a number of people to look at a couple of questions. What's the actual problem this germline editing can only sort of uniquely solve, given many, many alternatives, including somatic cell gene editing, gene therapy, and cell therapy? So what are the disorders that only you can get after? Because it seems like it's sort of a solution in search of a problem, kind of a false solution in search of a problem. And then what guardrails could be put in place to try to put downward pressure on such abuses, both scientifically and ethically? And we convened a lot of the types of organizations I talked about a few slides ago, patient organizations, actual patients. We had a great panel that Kevin was on, religious leaders, we had bioethicists, we had some of the leading scientists, including Kiran Musunuru at University of Pennsylvania, who Sadik was referring to as part of the ABKJ team, some of the kind of founders, early pioneers in gene editing. And we looked at, a few different things, right? We talked about the distinction between somatic cell editing and heritable editing. We talked about scientific and safety imperatives. We had a few scientists who said, look, I think you'd need to run, Keith Jung was one of them, one of the pioneers in the field, said, I think you'd need to have, 7 or 8 generations of non-human primate data that would show that this was safe. I'm not even sure how you could do that. And anyway, it would probably take at least 10, it might take 20, 25 years to generate that kind of a data set before you could even think about going and trying germline editing in human beings. There was a lot of discussion about ethical and societal responsibility, governance. Who's going to say what's in and what's out? Who calls balls and strikes? I mean, the FDA is not really competent to do that. People might say, well, I want to do things like, enhancement. Okay, well, there is no real great governance body, right? So there was a lot of discussion about that. And then there was talk of a call for really a moratorium while all this gets sorted out over many, many years. This is a very busy slide. We later put together a paper with our organizations that essentially did a number of things, but the upshot is we called for a 10-year moratorium on anything that looks like germline editing. It is like I said, it's sort of a false solution in search of a problem. There are many, many complex questions that need to be sorted out in the years ahead. Meanwhile, there's a lot of tremendous progress being made in somatic cell gene therapy, gene editing, and cell therapy that we think needs to be the primary focus. So we wrote that up in a paper, and we're going to continue to work on it. So with that, let me pause, and we can maybe turn it over to Kevin for questions.
Sure, we can pull back to the middle here and go ahead.
Oh, sorry.
So we want to definitely get a discussion going and open it up to our audience here and also our audience on Zoom. But let me start first by making sure that you two get the opportunity to tell us, what do you see is the biggest obstacle, the biggest concern that either one of you have in the near term? So let's look the next few years. What's the biggest concern that you have for this amazing technology and its possible positive or negative future?
I think just getting into rogue hands, building on what Tim was saying, Lori and I were talking earlier about CRISPR gene editing. Part of the power is how easy it is to use. I mean, middle school kids are now getting science lab classes about how to use CRISPR gene editing, and you can order a lot of the components online. And so I think there is that one component, but then also there are very few physician scientists like Becca and Kieran who understand the need from a disease perspective, and then also understand the capabilities of the technology and can deploy it in a safe and effective way. And I think that's the biggest concern that I have, is that it only takes one or two rogue actors to really ruin the field for everyone.
Yeah, I'll agree and build on that, and then introduce another one. So I do think the abuses are a real thing, and it's really interesting. Like if you think about technology, all sorts of technology over the last few decades, right? You always hear the sort of complaint, kind of understandably, that, and it could be related to things like intellectual property. It could be related to in a variety of fields. It could be related to IT systems and others. For many decades, it was a concern that, well, only the powerful they'll be able to control the technology, and therefore the masses are sort of subject to the whims and commercial desires of the very small number and the elite, and that's a huge concern. On the other side, as Sadik said, junior high school students are using CRISPR. It's a completely democratized technology, right? You can buy kits, you can do all kinds of things. But wait a second, so that's ideal. That's what we want, right? No, people are like, no, now everybody's got it. Hang on, the abuses could come from everywhere. So it is sort of like, wow, it is two dramatically kind of stark, different sets of concerns. I think they're both legitimate, right? But it is sort of, we always feared one, and then now we've got the other, exactly what we've always wanted, and there are legitimate concerns that people could abuse them, right? They call biohacking, where people are in their own garage and they could use this stuff. And every now and then you read these crazy articles about people. I dosed myself with CRISPR. Like, wait, what? Like you didn't run a clip? No, I just did it to myself. I mean, you don't see it quite as much today, but it was a big thing a few years ago. So it's wild what the technology can do. So I'm with Sadik on the abuses, and it's just not the U.S. It's sort of a global challenge. I think more proximate, and this is something I've been spending a little bit of time on the last few months, is we've got a sort of an ideological shift right here in the United States with the FDA, where their rhetoric has been extremely positive and supportive of cell and gene therapy. And they've rolled out, the FDA has rolled out a number of very important initiatives. The administration's rolled out a number of important initiatives. But at the same time, we've even seen this year a number of very promising cell and gene therapies have been sort of summarily shot down. And that's led to enormous frustration in the patient communities. Like I said, it's Rare Disease Week. There's a lot of discussion in Washington, D.C. this week because of that. One very promising program for Huntington's syndrome mostly impacts, I think it's about 50 boys in the United States. Really, really rough condition where time is life, time is functionality. There's some enzyme replacement therapies that are available, but they're limited in what they can do to slow the progression of the disease. And the FDA just shot down that promising gene therapy, much to the frustration of the patient community. There's a very important program for Huntington's disease that's coming up very quickly. People are very concerned that that may not get approved. There were a few others that were shot down by the FDA. So they're really dramatically raising the bar in real time. And that is concerning. And I think a lot of people are concerned, myself included, that this is a new sort of toxic methodological purity where they want to see the perfect clinical trials. So for example, they'd love to see randomized trials. Well, in some of these areas, the patient populations are too small. It'd be unethical to do that. You know, from an academic perspective, sure, you'd love to see perfect trials. But like I said before, time is life and time is functionality. These are imperfect medicines at this generation of where they are. They all have something that's not perfect about them. A friend of mine said to me this week, you'll get a kick out of this one, Kevin. We're talking about safety challenges and things. And he said, Tim, if they were perfect, we'd give them a different name. We'd call them miracles. We'd call them drugs for a reason. They're toxins that we're trying to use to slow the progression of the disease, or in the best case, potentially be curative, or at least have a durable response, but they're not miracles. So my concern kind of more near term, and it's playing out right now, as I've communicated to the FDA, and I wrote an op-ed about this just the other day. Right now with the FDA, it's a flashing red light today. This isn't something that's going to be played out in 6 months. It's a major problem right now. Yeah.
Okay, and as we heard, collaboration can be extremely effective in this regard. So we're going to do a little collaboration right now. We have Professor Seaman on the Zoom helping us out, and I'm going to ask Dr. Lavelle to help us out and grab the mic over there for our questions here in the audience. So we do a lot of collaboration among our Creighton faculty.
That's great.
So do we have any questions right here in the audience right now? Otherwise, oh, we got, thank you.
This will be a bit of a softball to get the conversation going, but
Like that.
should this be treated as a dose, as in big pharma, or a procedure that's going to be conducted at hospitals? And to use your analogy about doing CRISPR technology in your garage, I probably could remove my tooth in the garage if I had a cavity in it, but that's not something I'm going to elect to do because bad things could happen. Just thinking that, do we position this as a procedure where you go to the professionals to do it, or is this taking an Advil at home to reduce swelling or something? So,
Do you want to start, or you want me to? Your choice.
No, yeah, you start.
Yeah, I think it's an interesting. First of all, it's not a softball. This is actually a complex. It's a much more complex question. It's getting more and more debate and discussion, probably post baby KJ, than it did in the I don't know 4 or 5 years leading up to that. I would guess. And Sadik's in the middle of some of these discussions. I think this topic came up. I know some years ago, I raised it with a very senior FDA official who's no longer there, who's like, no, we have jurisdiction over this stuff for the very, very foreseeable future. You know, and yet it's reasonable for people to say, well, if I can, as Sadik said, if I can turn the flywheel, if I can get this stuff reduced down, couldn't it potentially be something where you could say it could be basically done in a clinic setting? I mean, people will say, well, geez, we do bone marrow transplants. We do a lot of other procedures and clinical surgeries and a lot of other things. People will say, well, what about genomic surgery, right? I remember when I was at Editas Medicine, so this is probably back in 2017 or 18, where I was out in California, I think at Berkeley, and there was a guy, a prominent academic who gave a big talk and said, we can do $1,000 genome surgery. I said, someone ought to make sure he's checking in with the FDA, because they're going to cart him out of here with handcuffs if he's not careful. And that disappeared pretty quickly until, kind of the more recent years. So I think the FDA will have jurisdiction over it for the foreseeable future. These are many, many times much more complex than a lot of the hospital procedures that have been around for a number of years. But I think this question is going to get asked more and more, and I think I could put it probably on my new list of
Yeah.
questions that are reasonable into the future, because as the science develops, you just don't know what the world could look like in the years ahead.
Yeah, and just to build on that, I mean, I think the two ends of the spectrum are it's a surgical procedure, and then you have an accreditation body. But in order to have an accreditation body, you need to have a field that's mature. Like surgery has been around for thousands of years, and we know how to simulate surgery. You know, I was taking a tour here of Creighton Labs. They have these models, that essentially training surgeons go there. How do you simulate molecular surgery? We don't have those models, and we don't have, the curriculums of medical schools don't really focus a lot on molecular intervention. It's really all about pathophysiological and anatomical intervention. So I think I can make an argument for it being regulated by the accreditation body, but for this to be a reality, you need to have a mature curriculum. You need to have folks that have been trained in the field that can train the next generation, and we're woefully away from that. So the alternative is to shoehorn what was done for baby KJ within the existing regulatory framework. And the challenges there are, as Tim says, historically we've regulated drug development on a risk-benefit basis. So I'm going to make a drug, and I'm going to look at the mean impact of this drug on the population. So net, if it's helping more people than it's harming, then I'm going to approve the drug. But a lot of those drugs, we don't necessarily know the mechanism of action. Like we don't know completely how Tylenol works, or Wegovy, Ozempic, these GLP-1s. Every day we're coming up with new bystander impacts beyond the diabetes. It's impacting on neural pathways. So a lot of these things we don't know the mechanism of action. As the value proposition with genetic editing or genomic intervention is you're getting to the root cause of the drug. So the benefit is significantly greater than taking a small tablet that can impact multiple organ systems. But the risk is being perceived as just as risky as a small drug. And I think we need to change that balance where the benefit is significantly greater potentially than the risk. But, we need to accelerate the development pathway so that the patients benefit from that intervention.
Yeah.
Thank you. Online,
question.
Professor Seaman, do we have anybody?
Yes, I have a comment from Father Clifton, so I'm going to read that, and then I do have a question as well. Thank you for addressing an exceptionally important topic in this Schweickert lecture. Thank you even more for the choice of panelists, both of who have summarized large, complex topics with amazing clarity and succinctness. Kudos to Dr. Schweickert for sponsoring this session and this series. So lots of positive comments. And then I would like to jump in with a question. One of my daughters just had twins a few days ago, due largely in part to technology and modern medicine through IVF. And so our family is knee deep in lots of conversation about embryos. And what do you see? You also mentioned the Chinese doctor that was put in jail, but sounds like he's out now. And, when we're asking the question to families, do you want some gene editing in order to have children, or gene editing in order to have a more healthy baby? Where is this line of what's legal and not legal? And who's going to decide that for not just our country, but globally? Do we have kind of a single point of truth where everything has got to be vetted in a certain way?
Whoa.
Yeah.
Well, good question. Let me try to unpack a few of those things, because there's a little bit of, I think in part the answer is a little bit of the science, a little bit of a little bit of the ethics meets regulation. I'm probably not going to remember all these things, and then and then the state of regulation today and the difference between traditional IVF versus this future where, like sort of like Gattaca, down the, you're not saying that obviously for today and for your family, but for down the road, the concerns about slippery slope and how do you put guardrails in place, right? So there's a lot there. So I think one thing I did not mention that's very important to mention for context is, to the best of our knowledge today, and the scientists to my left can speak to this more than I can. If people are thinking about germline editing, you're talking about a multi-generational effect. So the impact does not just die with the person that, first of all, does not exist today, but that so the life that gets brought, the person that gets brought into the world that has been edited as they are brought into the world, it will descend to all future generations. That includes any mistakes. So that gets into things like mosaicism, on-target and off-target events. So think of scientific mistakes that could have very severe impacts on the actual person, right? So first thing to remember is there is no person that's being treated. This is about, I want to bring someone into the world that has some traits or is minimizing some concerns in theory, right? But both the good and the bad will descend to future generations for many, many generations, as best we can tell, which is a big, big ethical thing. Very different than things like traditional IVF, right? So that's one thing. Two, coming out of like World War II, where we saw a lot of eugenic experiences out of Germany and things like that, certainly the European countries kind of banded together and said, we don't want this ever to happen again. And they put a lot of codes and treaties and things like that in place. Today, there's the Oviedo Convention, which was sort of born out of the spirit of those genetic abuses. I think that was put together in the 1990s. That clearly precludes across much of Europe anything that looks like germline editing. So it's long before we did anything in the US. Germany and the UK, as I recall, did not sign on to that. Germany didn't think it was strong enough. The UK thought it was too strong, but they both have kind of prohibited germline editing. And then you look across the globe in the US, China clarified that it's not prohibited there. Japan, many Asian countries. So it's really where there are laws in place that are in this neighborhood, I think they're exclusively to prohibit it for a lot of these reasons, concerns about abuse of the technology. So that's a little bit on the line. So anything that looks like it's going to edit or significantly manipulate an embryo tends to be swept up into these prohibitions. I know in the US, the FDA asserted jurisdiction in maybe the 80s, definitely by the 90s, for any manipulation of an embryo. They don't want it to happen in a fertility clinic. They're like, it has to come through the FDA, and we will require an IND, so a clinical trial. And Congress, in its wisdom, later weighed in and said, we're not going to allow that to happen in the United States. So it's very clearly precluded in the United States. Where else we go in? That slippery slope. Who decides? Well, right now, the public, as represented by their representative governments, in the case of democracies, and in the case of non-democracies like China and other places, the government has really decided that these are the guardrails that are in place, as kind of imperfect as they are. If you take that aside and just said, okay, well, geez, maybe someday, maybe the science could be ready, it gets into these next level challenges, right? Like things like, wow, well, what if my son, for example, has intellectual disabilities, right? Like, well, if someone had said to me once, you wouldn't want a world in which you're monkeying with intelligence. I said, well, I wouldn't want a world like that, but I know plenty of people that have kids with disabilities that have what we call lower IQs, and maybe they'd entertain it. So that could be, or people that, people say, well, what about height? Well, your perspective is a little bit different if you've got maybe dwarfism in your community. Some people would say, I would never want to touch that, but others might have a different perspective, right? Like, so it does get into this messy world of exactly that, who decides, and that rolls up to exactly, right? Somehow, at least we think of as in democracies, the public probably ought to have some kind of a say in this, and there probably would need to be some governance-like body, but this stuff has not been thought out, and that's another good reason why the technology, even if the technology were ready today, it's probably not ready for prime time, because we don't know how to answer a lot of these very weighty questions. I'm exhausted from answering this. I'm gonna let you guys take a piece of the action from there.
No, and I think to build on that, in terms of who decides, I would say one group that shouldn't decide, and I'm a scientist, but I think scientists shouldn't decide because I think any new shiny object, you want to be the first to deploy it and use it. And I think there's always that temptation. The incentive is for innovation, which is great. But here in this particular field, I intentionally highlighted that, we have the genetic roadmap, but half of the genes that we've mapped, we still don't really know what their function is. And then you have the epigenome, which is the layer that sits on top of the genome that affects different networks within the genetic pathways. So I think just we have to have that intellectual humility to know that intervening in a particular gene doesn't necessarily mean that it's going to lead to the downstream impact that you have. And I think scientists don't necessarily have that incentive to do that. I mean, it's good to do it in preclinical models, but this is where we need to engage the public more and really highlight some of the issues. And then, the other issue is, ARM does this really effectively, once or twice a year, going to the Hill, educating the lawmakers about the technologies and advocating. But I think we need to do more of that, and really it has to happen at the local level as well, state level, because a lot of fertility clinics aren't necessarily regulated by the FDA. There's a lot of opportunities for people to go off, and fertility has really enabled many lives and really improved many lives. But I think we do have this alternative universe, and it's been highlighted by publications like the Wall Street Journal, wealthy billionaires, essentially employing surrogates, 100 surrogates to carry their newborns. And I think, wherever there's an opportunity for a capability to be abused, I think even the guardrails that we have in place can always be bypassed by many individuals, and that could open up a Pandora's box. So I think it's kind of like a long-winded way of saying it's complicated, and I think it's still to be determined who's going to be intervening here.
But to put at least a small positive spin on this, I think also, just like there's all these threats, this also presents an opportunity.
Right.
And the opportunity is that if this technology does prove to be as promising as it could look right now, it does give us a leverage to go out to all the different groups in the world, all in their various silos right now, who aren't talking to one another in a constructive way, and arguing that we now need to get past those silos, we need to get past those barriers, we need to get outside of our little circles to have that difficult discussion as we continue to develop this technology, which has such tremendous promise for everyone. Again, sort of leaving the access question, off to the side for the moment. But that too is something that we certainly hope and discuss with our students in terms of, are we preparing them to be those people who can get up there and facilitate those discussions? Can they deal with the difficult patients and at the same time difficult communities? So I see some possibilities here that we, I think, have an opportunity to pursue. We have, yes.
Well, thank you so much for your time and for the topic. We can relate. My wife and I have a 42-year-old special needs son who lives with us, and we certainly can relate to that whole aspect of autonomy and where they go with that. But that aside, what I want to ask you is, to be really frank, how did you obtain informed consent with baby KJ for the therapy? Let me share with you why I asked that question. I'm a pediatric intensivist, so when I have a child in the PICU who is about to pass away and I'm going to do everything I can, consent doesn't even come into the issue, because whatever you're going to offer, they're going to say, where do I sign? So what are the ethics of informed consent for things like this, where you approach a patient who you have this to offer? What do you say? Or you just assume it's a slam dunk?
Yeah, it's a great question. So the treating clinicians would have the most detailed answer here, Rebecca and Kieran. But Children's Hospital of Philadelphia has been one of the leaders in pediatric interventions, and they have a lot of safeguards in place. But I think talking with Becca and Kieran, they were always very cognizant of not overpromising and not promising a miracle. And I think this field is really built on single patients and their families. Like, this field would not advance without folks being willing to take on that perceived risk for the field. And thankfully, the father of KJ had some familiarity with the field of genetic medicine. He's a tradesman by training, but he helped to build one of the facilities in the Philadelphia area that manufactures gene therapies, a type of gene therapy called an AAV gene therapy. So he was a bit familiar with the, but I think in this particular case, you look at the alternative, and this was the most severe of the mutations that one can have in the urea cycle disorder. So it literally was a life or death situation. And that's why they wanted to, they had identified other patients, but I think the ethical sort of it wasn't a black-white issue. Like, they could have survived longer and they could have waited for a liver transplant. Here, the situation was so stark that I think it really clarified those ethical boundaries. But you're absolutely spot on. I think as this moves to the front line, then the way that you communicate the benefit potentially and the risks, becomes really important, and having the IRBs being involved from the beginning.
Maybe just a cousin to that, not specific to baby KJ, but this comes up all the time in our world, because unlike a lot of other biotechnology interventions, if you treat a patient with a gene therapy, especially a child today, they might not be able to get treated again if something better comes along. And so now you're a parent where what the natural history is. Your kid's on the clock, right? And they're oftentimes the parents are watching the kid decline, and they are in this dilemma where it's like, this gene therapy could work, and maybe it's the first trial, right? So I might get the low dose, and it might not be enough to take care of my child, and then nothing else might be able to work, or maybe they're being treated with a high dose, and so throw the dice. Is it going to be toxic? I mean, we see this with toxicities, right? And deaths and serious side effects. What do you do? And I can tell you, having talked to some FDA folks, they see sometimes imperfect drugs coming along where the children are on the clock and they struggle with it, because they're like, I see something else 2 years out there, but it might be 3. And that's one other program. And if it stumbles, there's nothing coming along for a while. It is a major, major dilemma. So really tricky.
So we have one more. Oh, go ahead.
Go ahead, Father.
Oh, no, I was going to cut it off.
Oh.
Not yet.
Wow.
Yeah, that's right. He's got the mic.
Got another. They're all tough. These
Yeah, that's right.
I
They
got it.
are.
I actually got a couple of questions, so I really got to, I'll just ask one. What would happen today if the baby's not at CHOP or Hopkins or some other major academic medical center? A baby presents as did baby KJ, and let's say with this gentleman, he has a patient in his ICU, the pediatric ICU, who has a disorder similar to baby KJ. How might one get the sort of treatment that baby KJ had? I just think about some patients that I've had with some unusual stuff, not like this, but I've been very, very aggressive in getting them the best care possible. And, I get on the phone and make calls here and there. And, is that, what does the clinician do?
Well, I'll give the I'll give my quick to very blunt answer, and I'll let you
Yeah.
give the, a lot of times the answer is there's nothing we can do, right? This is the first time a personalized genetic medicine was kind of put together as a single test case with all this enormous team and
Yeah.
all this time. I mean, in the case of baby KJ, presumably they'd say, is this possible to do a liver transplant, right? What else, what other tools are there? But I mean, take the specifics of your recycle disorder aside and say it's some other intervention. I mean, you can sometimes get into a compassionate use situation. Depending on the situation, you can sometimes get into a clinical trial, but it's a real throw of the dice a lot of times.
And I think the journey starts with diagnosis.
Yeah.
And I think the diagnostic landscape in the United States is woefully inadequate and completely fragmented. And I think with a lot of these newborn genetic disorders, you're lucky if you live in certain states like California. They do newborn screening for certain inborn errors of immunity. So if you're born with a certain symptom, I mean, every baby gets screened there. In Florida, they screen for other things. In other states, they screen for other things. And I think the power of whole genome sequencing, so the price, now we have a technology that would enable rapid, 4-hour whole genome sequencing, not just the targeted newborn sequencing panel or NGS panel, but the entire genome. But I think the question becomes, how do we deploy that technology in a way that can be incorporated into the standard of care clinically? And how do we train physicians to be knowledgeable to read a lot of these reports? Because you can sequence the genome, but then, okay, great. What do I do with this?
Right.
You need to have some sort of actionable insight. And I think this is where AI can really be tremendously helpful, because it'll annotate and come up with actionable insights that can be validated rapidly, much more rapidly than having our fragmented ecosystem. And then final point here, other geographies like the UK and some of the Middle Eastern countries have invested in laying the infrastructure for newborn screening. And, they have like the, Genomics England, where their aim is to sequence a lot of babies. Because I think it's with increased sequencing and providing that access to diagnostic capabilities that you can then lay the infrastructure for subsequent intervention. And intervention doesn't have to be as extreme as like this personalized genetic medicine. It could be a change in diet. It could be some other, clinical intervention that's been tried and true. But just knowing what that genetic underpinning is what will help with the diagnosis.
Just another perspective on that, when we often have international gatherings and you bring that question up to ethicists from other parts of the world, there is no hesitation. They just tell the parents, your child's going to die.
Mhm.
And that's what they live with, right? Because that's their reality. So that, I think, is part of that global conversation that we have to have in terms of saying, do we have to live with that reality? Or is there a way in which we may need to say we have to live with less so that others can live with more? So all those things, I think, are going to be part of that immense global conversation that this could, in fact, engender. But as I was going to say, we do have another ethical issue here, and that is I did promise that I would not use them up completely. So give them at least a little reserve to enjoy the rest of the evening. So